Thank you very much, Margot, and thanks very much to the conference organizers to invite me to give this presentation. I'm going to talk about Mycoplasma ovipneumoniae, of course, in bighorn sheep, and try to update the conceptual model. And now my slide is not advancing. Oh, there we go. Here's the conceptual model that we published back in 2013. And my talk is going to be a little like this slide, probably hard to read, overly complex, and confusing, but I'll try to keep it on track as much as I can. This model sort of stipulates that the original origin of MOV is old world caprinae, domestic sheep and goats, which spill over to bighorn sheep, causing typically all age outbreaks. Survivors of those outbreaks may generally clear MOV, but there will be some carriers, and those carriers have an important role both in transmitting MOV onward to the lambs of the year, possibly for many years, and also to have infected animals that might move between bighorn populations, spreading MOV to new populations within a metapopulation. And then potentially in the end, this could go on for years, or it could result in MOV clearance and return to a healthy status. So what I'm going to start out doing is talk about a random set of individual observations or data or studies that reflect on different points within this schematic model that weren't available when the model was, that tended to support the model overall. And I'm going to talk about some new tools, genetic strain typing, and how those have reinforced other parts of this model. And then I'm going to talk about confusions and contradictions, which test some doubt on parts of the model or move outside the model. And lastly, the main topic will be talking about possible alternative causes of pneumonia that don't involve MOV. And I'll close up with a brief list of bullets of what I think are research priorities going forward. So one of the predictions of the model is that when MOV is introduced into a naive population, disease results fairly shortly thereafter. And as people have studied bighorns more and monitored their herds better, these have become more precise. And here's an example that happened a few years back at the National Bison Range bighorn sheep operation. These sheep had been sampled periodically for various reasons, and testing for MOV was included between 2006 and 2015 with a sort of an emphasis in 2015. All of these tests, both antibody and PCR, were negative. Then at the end of the 2015 biological year, the start of the 2016, all of a sudden disease was observed in this population. And from the very first dead sheep, 90% of the sheep that were tested had MOV infection detected by PCR. So the model predicts that disease would follow shortly after introduction, and herds like this show that that has followed accurately. So in addition to introduction, the model talks about persistence of these shedders, and particularly persistence of shedder ewes, and an important role of those shedder ewes in transmitting to lambs, causing lamb pneumonia and lamb poor recruitment. And this slide depicts some captive studies at Washington State University, and Logan Wayand is leading this and is preparing the paper that will describe it. But briefly, it was a group of 6 ewes that had undergone MOV-based pneumonia outbreak, or that had survived the MOV-based MOV pneumonia outbreak, and others died. But of these 6, by a year after the outbreak, 5 of the ewes, I used B through F in this slide, had cleared MOV, but good old slide A, who is my title slide, sheep with a snotty nose and all, remained MOV positive for a couple of years after this. And we were able to use this situation to try to test the role of this carrier ewes in transmission to lambs. And so we call this a carrier segregation experiment. In the first year of this study, we took our carrier A and put her in a pen with 1 non-carrier and put the other 4 non-carriers in a separate pen. And the outcome that year was 2 lambs born in the carrier pen, both got pneumonia, both died, 4 lambs born in the non-carrier pen, and all stayed healthy. For the second year, we just basically crossed over some of the non-carrier ewes, put a couple more in with UA, and took UD and put her in with the non-carrier pen. And the outcome is just as the model predicted. The 3 lambs born to the ewes in the carrier pen all developed pneumonia and died. And the 2 lambs that were not stillborn in the non-carrier pen remained healthy. Francis Cassier's talk later on is going to talk more about this study and related studies. So in addition to persistence of these carrier ewes in captive studies, you can, it turns out, also address it in the field. And this has taken the form of test and remove studies. And I'm going to just show this one slide with some data from Tyler Garwood's paper that was published this year. But basically, Tyler identified a treatment herd and a control herd. The treatment consisted of removal of documented MOV carriers and the control not removal. In his studies and repeated samplings, Tyler identified 2 chronic carriers each in the control and the treatment herds. And this slide just shows the data the first year after the chronic carriers were removed. In the treatment, which is the left pie chart within each age group, no pneumonia was observed, and the pneumonia persisted in the control groups. And that was true for both adults and for lambs. And again, Francis Cassier is going to talk later today about more test and remove studies. But as predicted, if you remove the carriers and remove the source of MOV in the herd, the lamb disease stops. This can also happen without human intervention. And this slide shows the results from that national bison range outbreak, which I talked about earlier in terms of disease onset shortly following disease MOV introduction. But in this case, we're looking at after the disease outbreak. So the pneumonia epizootic was in biological year 2016. And as the herd was followed thereafter, after the 85% die-off, the following year, nearly all animals were antibody positive, but substantially fewer animals were PCR positive. It stayed low in 2018. And at the end of 2018, the last known carrier in the U group at the NBR died. And so MOV cleared itself, and while lambs died of pneumonia in biological years 2016, 2017, and 2018, in 2019 and now 2020, the lambs still died for various causes, but some survived, and there was no pneumonia observed at all. So this is an example of the situation where the sheep took care of themselves and cleared the MOV, and the disease followed shortly after the loss of the last MOV carrier. I think these studies that show the cessation of disease following removal, either spontaneous or planned, of MOV are really the strongest new evidence that's come up since we proposed the hypothesis, because action was taken or things happened just to one organism of the many pathogens that are carried, and yet the disease situation changed dramatically. I'd like to present a few slides about genetic strain typing of MOV, because it's been a tool that's been very useful in identifying or testing various other parts of the conceptual model. So we use a genetic strain typing tool called MLST, multi-locus strain typing, where we basically have identified 4 segments of the chromosome that are very informative and yet conserved enough we can amplify them, we can sequence them, and we compare the sequences and identify strains that differ based on different sequences in these 4 conserved regions. This is a figure from Pauline Kamath's paper from last year about this very method and its results. And the slide shows results from domestic sheep and from bighorn sheep. The very steep, this is a rarefaction curve. So as you test more and more MOV samples, how many more strains do you detect with this method? The blue line depicting domestic sheep shows a very steep increase. Almost every new strain tested result, every new detection tested resulted in a new strain. Tremendous diversity of MOV within the domestic sheep. In contrast, the bighorn sheep populations, it flattens out very fast. And so most bighorn sheep populations just have one or a few strains of MOV present. So a couple of implications of this. First of all, it supports the conceptual model because we hypothesized that the domestic sheep were the reservoir, and you would expect more diversity in the reservoir than in the spillover population target. And second, that because the diversity is so high, finding matching MLST strains by chance alone is generally very unlikely. So finding a match pretty much usually indicates transmission of the same strain. So this strain typing method has been applied in a number of situations. I already mentioned Pauline Camus' paper, but this shows some of the strain type results from the Idaho populations, the Hells Canyon in the upper left, and the central Idaho population in the lower right, and the strain types are color coded. And so in the Hells Canyon area, that light blue strain, you can see there's a number of dots, and that those dots each represent a population, and the blue indicates a strain of MOV that has been spread from amongst the various populations in this metapopulation, probably by bighorn sheep moving it from one to the other. And the same for the central Idaho population with the yellow and pink strains likely being transmitted amongst those populations by carrier bighorn sheep. Genetic strain typing has also been very helpful where there was MOV present before and after an event in explaining what happened with that event. And so here's a slide based on data from Hells Canyon from Francis Cassier's population. And as you can see in the timeline in the lower right, this blue strain, the dots, have been present since 1995 and detected pretty much every year in a number of these populations within the Hells Canyon metapopulation. And it's the strain that's been most associated with repeated annual outbreaks of lamb pneumonia year after year, while the adults generally stayed normal and healthy looking. In 2013, however, all of the adults in one population and in the Black Butte population suddenly became ill, and clearly ill with coughing and pneumonia, and some of them died. This would have been quite a mysterious event because MOV had been present before and after, and what was causing all these sheep to suddenly die, until we did the strain typing and we realized that a new strain depicted in red on this graph had been introduced into this population and had spread through the adults. So this clarified an important point regarding Mycoplasma ovipneumoniae and immunity, in that immunity seems to be strain specific. So being immune to one strain doesn't protect you from being infected with a new strain. And it really, without strain typing, this would have been a mystery disease event in the presence of stable MOV. And that strain typing gave us an insight into what was really happening. This reflects a bigger problem in many, especially remnant native herds, where multiple strains are involved. As those strains circulate or as new strains are introduced, it can be very difficult to suss out what's happening. We've also been able to use genetic strain typing to help support the first part of the conceptual model, which is the spillover from domestic sheep, domestic goats to wild sheep in this. And this slide depicts two examples that both were developed by the folks in Nevada. On the left, the strain. First of all, these figures show phylogenetic trees. There's not much of a tree in either of these two examples because all of the strains as connected by the vertical line are identical, and so they're the same strain type. On the left, it shows strains identified from a half dozen of the snowstorm bighorn sheep that died of this in this epizootic, along with a strain from a single domestic sheep that was found straying within the snowstorm mountains bighorn range at the same time. And so that domestic sheep had the identical strain as the outbreak, and is the presumptive source of that outbreak. It's certainly more parsimonious to believe that the domestic sheep brought it into the bighorns and it then spread amongst the bighorns than vice versa. On the right is a more unique example of two bighorn sheep and a single domestic goat. These three animals were found commingled but separate from the bighorn range. And as often happens, the animals were euthanized to prevent risk to if they return to their bighorn population. And again, these three animals all had identical MOV strain type in them, and both bighorn sheep had pneumonia as documented at necropsies. And again, the domestic goat here is the presumptive source of that strain of MOV. It was a domestic goat clade of MOV determined by the strain type that had spread to the bighorn sheep and caused this pneumonia. This is a third example that happened more recently. After a severe pneumonia outbreak in a bighorn sheep population, in trying to determine what the possible sources for that MOV introduction, 2 domestic sheep operations were identified that were most closely adjacent. After consulting with the owners of these operations, we got permission to sample both operations for MOV to look at strain types to see if any matched the bighorn sheep outbreak. Both Operation 1 and Operation 2 had diverse strains of MOV present. However, in Operation 1, there was none of these strains matched the bighorn sheep outbreak strain. In contrast, in Operation 2, despite the diverse strains present, we found 6 ewes that carried the bighorn sheep outbreak strain. And so clearly Operation 2 was identified as a presumptive source, or the most parsimonious explanation for the source of the MOV spillover that affected bighorn sheep. In this case, it was the bighorn sheep that wandered onto the domestic sheep operation almost for certain, because the sheep were confined there by fences, and then those bighorn sheep probably carried it back to the population. So this really does tend to support the concept that strains of MOV pneumoniae from the reservoir spill over to wild sheep and infect them and cause pneumonia. So the next part of this talk is going to switch to confusing and contradictory things. And I've just got a few items I want to highlight here. There's a lot more confusion and contradictions out there I won't have time to talk about. But one that's received some attention in recent years is personal communication from Kimberly Beckman, and also a publication by Margaret Hyland indicating the presence of Mycoplasma ovipneumoniae in caribou in Alaska. And this is a figure from that Kimberly provided showing the caribou ranges. And all of these dots, other than the purple ones, reflect prevalence of animals sampled which were found to be carrying Mycoplasma ovipneumoniae by 1 or more laboratories. So these are, the colors here are not strains, they are which labs detected it, but these were strain typed wherever possible on the strains that were detected through wattle. And in fact, all of these populations in which MOV was detected, where it was possible to get a strain type, it was an identical strain type across all these ranges. And perhaps not surprisingly, similar sampling in Dall sheep in Alaska detected Dall sheep carrying Mycoplasma ovipneumoniae in a number of these populations, several of which were pretty close to the caribou ranges, and all of which had, again, exactly the same MLST strain type of Mycoplasma ovipneumoniae. So very unusual situation, a single MLST MOV strain type in these 2 wildlife species, one being a non-Caprinae, which is the dogma had been that MOV is host specific to Caprinae. And I think it still generally is, but here is clearly a strain that is not host specific. Also unusual is the broad geographic range of a single strain. In wildlife in the lower 48, well, I'll talk about an exception, but generally we don't see strains spread this far. And in both host species here in Alaska, to my knowledge, there's no compelling evidence of disease association. This MOV seems to be circulating in both species with minimal or no adverse health effects. This is weird enough, but then this year, and this is really what compelled me to add this to this talk today, the Wyoming Wildlife Lab and Dr. Hank Edwards identified a sample from a white-tailed deer with pneumonia that had Mycoplasma ovipneumoniae. Hank sent samples from the animal to Wattle to do strain typing, and Wattle was able to do the strain typing and identified strain BHS 37, which was a strain type named by Pauline Kamuth in her paper. And this strain actually received quite a bit of discussion in Pauline's paper because it was the sole exception of an MLST strain that was very broadly geographically distributed. And it has been detected previously in bighorn sheep in 4 states and in a mountain goat in a 5th state. So parallels with the Alaska situation are compelling to me. A single MOV pneumonia strain, MLST strain in multiple species, including a non-caprinae, white-tailed deer, across a very broad geographical extent. And despite the fact that it was detected from a white-tailed deer sample, I don't think at this present time there's any compelling evidence of a disease association. And by the way, the 4 bighorn populations in which this strain of MOV has been detected, this strain is also one of those ones where there's not really a compelling disease association because each of these populations had more than one strain type, and most of the disease samples from those populations had been a different strain type and not BHS 37. So these studies raise a lot more questions than they answer. I don't have those answers, but there are some really interesting compelling parallels. Okay, I'd like to really switch gears now and talk about the possibility of other causes of epizootics of pneumonia that are unrelated to Mycoplasma ovipneumoniae. It's been surprising to me since we proposed this hypothesis that we haven't, how many or such a high percentage of the known outbreaks that have been worked up have included plausible or definite involvement with Mycoplasma ovipneumoniae. The best documented example of one that doesn't is one that's being under investigation right now in California. San Gorgonio desert bighorn sheep had an outbreak identified in early 2019, clearly had pneumonia, significant population decline, but of the 13 animals sampled, there was only a single MOV pneumonia detection, and it was a strain type that's not particularly disease associated. So in my view, this represents a significant pneumonia epizootic that's likely not MOV pneumonia related. And we'll have to wait for the local investigators to continue their work to try to explain what did cause this outbreak. Hopefully more information will be forthcoming. That sort of brings me to some captive studies that we have done that also raise the possibility of epizootics of pneumonia unrelated to Mycoplasma ovipneumoniae. And I'd like to start out by going back to this paper from 2012, in which we pointed out that when bighorn sheep had been commingled with domestic sheep in a number of publications in the previous years, overall in those publications, 98% of the bighorn sheep were dead within 100 days, generally of epizootic pneumonia. And in comparison, this dotted line above shows what happened when 50 or so bighorn sheep were commingled with non-domestic sheep animals of various species, much lower death loss. And what we did in this study was we took 4 bighorn sheep and commingled them with 4 domestic sheep, but these were domestic sheep that we had demonstrated were free of MOV by repeated testing. And in that situation, nothing happened for 90 days. 1 bighorn sheep died at 90 days, but 3 out of 4 survived, very different than the 98% that had died in the previous studies. Interestingly, that one that died had plausibly a pure Mannheimia hemolytica pneumonia, the agent that had received a lot of attention for bighorn sheep pneumonia in past years. And I think this study raised the question in our minds, and Vic Coggins in particular kept bugging me to follow up with this. Can prolonged contact with MOV negative domestic sheep also have adverse health effects on bighorn sheep? So I'd like to quickly describe 2 experiments we did in 2018 and 2019. In 2018, we took 6 best bighorn sheep and commingled them with 3 MOV pneumoniae free domestic sheep and watched them for a long time. And nothing happened at 100 days. We had to remove one of the bighorn sheep for another cause. After 111 days, there was no pneumonia in that animal, and obviously no ovipneumoniae. But at 119 days, we had 2 additional domestic sheep that originated from the same premises as the first 3, also MOV pneumoniae free, which we added to the pen. And that had pretty striking results in that all of the bighorn sheep in that pen died between 19 and 50 days later. All of them died peracutely, sick one day and a half a day or a day later dead, or had to be euthanized. And all of them had Mannheimia hemolytica as the predominant growth in their lungs. These were not generally polymicrobial pneumonia, as we see with Mycoplasma ovipneumonia. They were pure culture with Mannheimia hemolytica and pure population. In 2019, we did a partial repeat of this study. We took 3 other bighorn sheep, commingled them with the same 2 killer domestic sheep that we'd added at 119 days in the first year. At day 10 of this study, after commingling, twin bighorn lambs were born to one of these bighorn sheep ewes. And those lambs died within a couple days of birth. And then 2 of the bighorn sheep that were commingled originally died at 62 and 105 days after commingling with these 2 bighorn sheep. And all these animals, both lambs and adults, had very peracute deaths, of which Mannheimia hemolytica was the predominant bacteria in the lungs. And the one surviving bighorn sheep was removed when we terminated the experiment after 170 days. So I think these studies document epizootic Mannheimia in bighorn sheep commingled with domestic sheep. It's odd that the apparent association was with specific individual domestic sheep, the two sheep that we added second in the first year, not necessarily the duration of commingling. And all 5 of these domestic sheep had Mannheimia hemolytica carried in their pharynx. All of them were positive for the leukotoxin gene, which is the virulence factors pre-commingling. So they didn't differ in the carriage of Mannheimia hemolytica as far as we know. So I think this study does raise the potential that non-Mycoplasma ovipneumonia, or maybe specifically Mannheimia hemolytica bighorn sheep die-offs may occur after contact with domestic sheep, or possibly with other carriers of these bacteria. The fact that we haven't seen outbreaks that appeared like this in the wild makes me think this is not a common happening, and that sporadic disease might be much more common, but I thought it was worth mentioning here. My very last slide is a few outstanding issues that I think are high priority for further research. One is just being able to distinguish more virulent and less virulent or non-virulent MOV pneumoniae strains would be outstanding help in sorting out these populations where multiple strains are present, especially in those unusual but not rare populations where MOV pneumonia is constantly present but disease is not seen. We also know that epidemiological factors, age at exposure, exposure dose, season, and host factors impact disease outcomes, but we don't really know exactly how this happens. And particularly for variation in severity of lamb pneumonia from year after year, we think these factors might be very important. We might be able to work that out through some targeted studies. And the last outstanding issue, which I haven't talked at all about today, which I think is really key to reducing risk to bighorn sheep in the long run, is research to control or eliminate MOV pneumoniae from domestic sheep and goats to reduce risk to wild sheep. So lots of folks to prepare, and these lists are probably incomplete, but I'd like to thank all the funders and all my co-authors. That's what I prepared for today. Thank you very much.
Thank you, Tom. Actually, you've got 1 minute to spare. So let's take one quick question from the Q&A. And I would remind, as was sent out earlier, that it is the Q&A that you put your questions into and not the chat line. But I think maybe a quick one with a question coming in: have multiple strains of MOVI been detected in the same animal?
We have detected multiple strains of MOV in the same animal in both domestic sheep and in bighorn sheep. You generally don't detect them at the same time because the strain typing method as used right now, strain types the more predominant of the multiple strains that are present.
Okay, perfect. I think we'll leave it at that. Certainly you've provided a wonderful platform and overview of what we know, in some cases what we don't know, and left us thinking about what we would like to know, which is a perfect opening to a plenary session. So if we'll move on to the next presentation, Doug, if you could move forward and share your screen.